Hello everyone. I am Mukesh, a second year MBBS student from GMCS Sundargarh. Today I am going to discuss glycolysis, a topic from carbohydrate metabolisms. Glycolysis is less important as compared to gluconeogenesis for the university examinations, but for understanding of the gluconeogenesis, understanding of glycolysis is play important roles. So let's discuss about the glycolysis. Glycolysis is derived from the two words, that is glycose and the lysis. Glycose means sweets or the sugars, where the lysis is splitting. So splitting of carbohydrate to pyruvates or the lactic acid is called glycolysis. Site of the reaction is cytoplasm and it takes place in all the cells. And if you see about the steps of glycolysis, glucose is converted into glucose 6 phosphate with the help of hexokinase or the glucokinase, where these two enzymes plays an important role. Glucokinase is insulin dependent, where hexokinase is insulin independent, and generally glucokinase acts when blood glucose level is greater than 100 milligram per deciliter, but hexokinase acts at a low glucose concentration. That's why hexokinase has less Km as compared to glucokinase. That means enzyme specificity for the hexokinase is better than the glucokinase. Okay, so here the one utilization of ATPase kinase was utilized, and we know that when kinase is present, there will be the utilization of magnesium ionized coenzymes. Okay, so glucose is converted to glucose 6 phosphates, and this is isomerized to fructose 6 phosphate with the help of isomerase, and fructose 6 phosphate is converted into fructose 1,6 bis phosphate with the help of phosphofructokinase, and there will be the utilization of 1 ATP and magnesium ionized coenzymes. So fructose 1,6 bis phosphates is converted into glyceraldehyde 3 phosphates and dihydroxyacetone phosphate with the help of aldolase A. Here is the, you will see about the glyceraldehyde 3 phosphate. This is the actual structure of glycerol, and if you add one phosphate at the third position, it will convert into the glyceraldehyde 3 phosphates. Similarly, if you see about the dihydroxyacetone phosphate, where is the ketone group is present, that's why dihydroxyacetone phosphate is mostly, there will be the two hydroxy group at the first and third position and one ketone group or the functional group at the second carbon. That will become dihydroxyacetone phosphate. Dihydroxyacetone phosphate is converted into glyceraldehyde 3 phosphate with the help of isomerase enzymes, and after converting into glyceraldehyde 3 phosphate, glyceraldehyde 3 phosphates undergo glyceraldehyde 3 phosphate dehydrogenase, gives rise to 1,3 bis phosphoglycerates. Here will be the, as you know that this is aldehyde 1 and this is the acid 1. That's why we will convert the aldehyde to acid by the known as oxidation reactions. That's why we will utilize the enzyme, that is glyceraldehyde 3 phosphate. Here NAD plus is the acceptor molecule which accept 1H from this glyceraldehyde 3 phosphate and convert into 1,3 bis phosphoglycerates, and 1,3 bis phosphoglycerate is converted into 3 phosphoglycerate by the removal of one phosphate group from the 1,3 bis phosphoglycerates, and this phosphate group is accompanied with ADP, and there will be the formation of ATP. This ATP is formed from the 1,3 bis phosphoglycerate to 3 phosphoglycerate is known as substrate level phosphorylation, as there is no oxidative level phosphorylation. That's why it is known as the substrate level of phosphorylation, where higher energy state is converted into lower energy with the release of ATP. Okay, similarly 3 phosphoglycerate is converted into 2 phosphoglycerate with the help of enzyme mutase, and these 2 phosphoglycerate is converted into phosphoenol pyruvate with the enzyme of help enolase, and there is again presence of the magnesium ions. There is a common clinical importance of enolase in case of blood sugar estimations. Okay, generally we use the fluoride vials for the blood collection that inhibits that competitively with the enolase. That's why there will be no utilization of glucose and from the blood vials. That gives a correct result for the blood sugar estimation. Similarly, this 2 phosphoglycerate is converted into phosphoenol pyruvate, and with the help of enolase there will be the release of H2 molecule, and this phosphoenol pyruvate is converted into pyruvate by the help of pyruvate kinase, and here again kinase is present. There will be the relation with ATP, that is the production of 1 ATP, and magnesium ion is utilized. Okay, this is called actually aerobic glycolysis, and if pyruvate is converted into lactate with the help of lactate dehydrogenase, this undergo anaerobic glycolysis. Okay, and there will be the utilization of NADH to NAD plus. So this is actually the steps of the glycolysis. For the memorization point of view, no need to memorize each and every enzymes, just memorize the main enzyme like glucokinase or the hexokinase. Similarly phosphofructokinase, there is aldolase and glyceraldehyde 3 phosphates. and the pyruvate kinase. If we say about the steps which are important for the regulatory step of glycolysis, that is the first step, which is on the irreversible one. Similarly, the third step, that is phosphofructokinase mediated, and the 10th number step, which is the pyruvate kinase mediated. These are the rate regulatory steps for the glycolysis, and this mostly regulate the process of glycolysis. Okay, this is all about the steps of glycolysis. Just memorize these steps, and no need to memorize with the help of mnemonics, that may create a confusion during writings. That's why better is to practice by writing one or two more times. Okay, so let's see about the significance of glycolysis. Significance of glycolysis, we say about it is the only pathway that is present in all the cells of the body. That means glycolysis is the only pathway of energy respiratory pathway, respiration, in which there will be the production of energy in all cells. Okay, only source of energy in the erythrocytes. That means in erythrocytes, as we know that the lack of mitochondria, that means there will be no oxidative phosphorylation. That's why the source of energy is only glycolysis. During strenuous exercise, anaerobic glycolysis form the major source of energy. Let's understand this statement. During strenuous exercise, anaerobic glycolysis form the major source of energy. As you know that in strenuous exercise, there will be the oxygen demand is increased, and that's where, because of oxygen demand increase, there will be less oxidative phosphorylation, and less oxidative phosphorylation leading to less production of NAD plus, that leads to decrease in the glycolysis. So if NAD plus is decreased, that means these steps of glycerol 3 phosphate dehydrogenase will be inhibited, and glycolysis may not operate properly. That's why in anaerobic glycolysis, here the NADH is converted into NAD plus, which gives rise to constant production of NAD plus. That's why in strenuous exercise, NAD production is produced by the anaerobic glycolysis. That's play important role for the strenuous exercise and anaerobic glycolysis. Synthesis of non-essential amino acids and the glycerol part of the fat is also synthesized from this role. Okay, if we'll say about the energetics of glyceraldehyde. Energetics of glyceraldehyde, as here the one NADH product from 3 glyceraldehyde. Similarly, from dihydroxylation there will be 2 NADH produced, and 1 ATP from this step 7th, and the 1 ATP from the 10th step. Similarly, there will be the utilization of 1 ATP from the first step and the third stage. That means if we will do anaerobic glycolysis, that means there will be the 2 NADH and 2 SLP from 1 glyceraldehyde 3 phosphate, another SLP from 2 SLP from the glyceraldehyde 3 phosphate. That means there will be the production of 5 ATP from NADH, SLP2 and another SLP2, gives rise to 9 ATP. As you have utilized 2 ATP for the in energy utilizing step, that means net ATP in the aerobic glycolysis is 7, and in case of non-aerobic glycolysis there will be the 2 ATP, as NADH is not useful in the anaerobic glycolysis. Okay, so if you say about the regulation of the glycolysis, regulatory enzyme that irreversible step, that is glucokinase, hexokinase, phosphofructokinase and pyruvate kinase. If you say about the hexokinase, this is regulated by the feedback inhibitions. That means feedback inhibition is something like what? Glucose is converted to glucose 6 phosphate. If the level of glucose 6 phosphate is high, that's negative feedbacks to the hexokinase or the glucokinase. There will be less production of the glucose 6 phosphates, and if there will be decrease in the glucose 6 phosphate, that will give a positive feedback to hexokinase and increase production of the glucose 6 phosphate, leading to increased glycolysis. Similarly, in phosphofructokinase, ATP and citrate inhibitors, that means phosphofructokinase is the rate limiting step for the glycolysis, and in presence of ATP, that means we have no energy. That's why they inhibit the glycolysis process, and AMP, that is low energy state, induces the phosphofructokinase. And similarly, we say with the pyruvate kinase, that inhibit by the ATP, that means energy state no requires glycolysis. And if we say about the hormonal regulation, that insulin favor glycolysis by inducing that three rate limiting that three regulatory enzymes, and glucagon and glucocorticoids inhibit glycolysis. That means they, as we know that they are hyperglycemic hormone, that means they wants to increase the glucose, they will reduce the glucose utilization by the peripheral system. That's why they inhibit the glycolysis. Thank you everyone.